We are going to start with the chapter number 2 of biological classification of class 11th. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis of classification. Aristotle was the earliest to attempt a more scientific basis of classification. He used what? Simple morphological characters, again important line, to classify plants into trees, shrubs, herbs, and he also divided animals into two groups: those which have red blood and those which do not. Those which had red blood, they are called anema, and those which do not have, they are called anema. Okay, next is yes, in Linnaeus time a two-kingdom system of classification with Plantae and Animalia kingdom was developed that included all plants and animals respectively. This system did not distinguish between what? This system did not distinguish between eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic and non-photosynthetic organisms. Classification of organisms into plant animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, yes, evolutionary relationships, etc. Classification systems for living organisms have hence undergone several changes over the time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups or organisms be included under these kingdoms have been changing, and the number and nature of other kingdoms have also been understood differently by different scientists. So, this is the table of 5 kingdom classification which was given by R.H. Whittaker. Okay, now moving ahead, we will come to the table later on, but first let us come to let me change the color first. Let us take green. Okay, R.H. Whittaker proposed a 5 kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification, this is important line, be careful. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. The 3-domain system also have been proposed that divides the kingdom honor into 2 domains, leaving the remaining eukaryote kingdom in the 3rd domain, and thereby a 6-kingdom classification. This was given by Carl Woese. Okay, but this is not mentioned in NCERT, but you should know. You will learn about this system in detail in higher classes. Okay, let us look at the 5 kingdom classification to understand the issues and consideration that influence the classification system. Earlier classification system. Now, earliest classification system included bacteria, blue-green algae, fungi, mosses. Sorry, ज़्यादा डार्क हो गया. Included bacteria, blue-green algae, fungi, mosses, and ferns, gymnosperms and angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cell. Yes. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae. An important line. It brought together the prokaryotic bacteria and the blue-green algae with other groups which are eukaryotic. It also grouped together the unicellular organism and the multicellular organisms, for example, Chlamydomonas and Spirogyra. They were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants. They also showed a characteristic difference in their wall composition. The fungi had chitin and the plants have wall made up of what? Cellulose. Yes, moving ahead. In their walls, while the green plants had a cellulosic cell wall, when such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic were grouped together under kingdom Monera, and unicellular eukaryotes, they were grouped in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, and Paramecium and amoeba, which were earlier placed in animal kingdom, and earlier algae was placed within plants. Okay. It has put together organisms which in earlier classifications were in different kingdoms. This happened because of the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only morphological, physiological, and reproductive similarities but also phylogenetic, that is based on evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, fungi of Whittaker's system of classification. The kingdom Plantae and Animalia commonly referred to as plant and animal kingdoms respectively. Now, we are starting with kingdom Monera. Kingdom Monera. Bacteria are the sole members of kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in the handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans, where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on the shape: the spherical coccus, and the rod-shaped bacillus, and the comma-shaped is the vibrio, and the spirillum. Okay, these are the shapes of the bacteria as cocci, bacilli, spirali. So spiral shaped bacteria have flagellum, yes, vibrio. Now moving with cocci bacteria, example is Streptococcus pneumoniae. Okay, moving ahead. The bacterial structure is very simple. They are very complex in behavior. This is a very important line. Compared to many other organisms, the bacteria as a group shows the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms or on dead, decaying organic matter for food. Starting with the archaebacteria. These bacteria are special since they live in the most of the harsh habitats such as extreme salty areas. We call them halophiles, or they live in hot springs. They are called thermoacidophiles. In marshy areas, they are called methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure. Another important line: this feature is responsible for their survival in extreme conditions. Methanogens are present in guts of several ruminants such as cows, buffaloes, and they are responsible for the production of methane from the dungs of these animals. ठीक है, moving on for eubacteria. Eubacteria is true bacteria and archaebacteria is older. There are thousands of different eubacteria, true bacteria. They are characterized by presence of a rigid cell wall, and if motile, they have a flagellum. Flagellum is made up of protein flagellin. Okay. The cyanobacteria is also referred as blue-green algae, have chlorophyll A similar to the green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial, and filamentous. Another important line: they can be freshwater or marine or terrestrial. The colonies are generally surrounded by gelatinous sheath or mucilaginous covering. They often form balloons in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cell called heterocyst. ठीक है. Example is nostoc and anibina. The chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia. ठीक है. Use the released energy for ATP production. Yes, another important lines. Yes. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk. That is lactobacillus. And another is production of antibiotics fixing nitrogen in legumes. Yes, this diagram of the nostoc is given. Nostoc is what it is photosynthetic autotroph. ठीक है, which is an example of cyanobacteria. देखो, this is heterocyst. In this nitrogen fixing occurs, mucilaginous sheath. Okay, and they often form blooms. Moving next to, yes, आपने को we have nitrogen in legume, roots, etc. Some are pathogen causing damage to human beings, crops, farms, and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes under unfavorable conditions they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA. DNA transfer from one bacterium to other. Yes. Now the next is the mycoplasma. Again, this one paragraph is very, 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 very important. The mycoplasma, they are the organisms. They are the organisms that completely lack the cell wall, and they are the simplest living organisms known and can survive without oxygen. They are aerobes but can survive without oxygen, and many mycoplasma are pathogenic in animals and plants. They are PPLO, pleuropneumonia like organisms. Now moving ahead for kingdom Protista. For this I will change the color. Yes. Okay, so yes, all single cell eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. But maybe a photosynthetic protestant to one biologist, maybe a plant to another. In this book, we include crucified dinoflagellates, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the others, dealing with plants, animals, and fungi. Being eukaryotes, a protestant cell wall contains a well-defined nucleus and other membrane-bound organelles. Important line, that being eukaryote, they are unicellular eukaryote. The protestant cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. Moving for chrysophytes. Yes, the group includes diatoms and golden algae. Diatoms and desmids are. Yes, they are found in freshwater. They are found in freshwater as well as marine water environment. They are microscopic and float passively in water currents, that is plankton. Most of them are photosynthetic. In diatoms, the cell wall form two overlapping shells which fit together as a soap box. The walls are embedded with silica, and thus the walls are indestructible. Thus, the atoms have left behind large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producers in oceans. Now, moving ahead for what dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigments present in the cell wall. The cell wall has stiff cellulose plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the polyplates. Very often, red dinoflagellates, example Goniolax, undergo such rapid multiplication that make the sea appear red. A toxin released by such large numbers, they may even kill other marine animals such as fishes. This is the diagram of dinoflagellates. Now moving for euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle, which makes their body flexible. They have two flagella, a short and long one, though they are photosynthetic in nature in the presence of sunlight. When deprived of sunlight, they behave like heterotrophs by predating on other small organisms. Yes, interestingly, the pigments of euglenoids are identical to those present in higher plants, chlorophyll A and C. Yes. Example is euglena. Euglena is what? Example of euglenoid. Now moving for slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves engulfing organic material. Under suitable condition, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable condition, the plasmodium differentiates and form fruiting bodies bearing spores at the tip. The spores possess true walls. They are extremely resistant and survive for many years, even under adverse condition. The spores are dispersed by air currents. Now moving for protozoans. Protozoans are heterotrophs. Protozoans are heterotrophs. Heterotrophs and live as predators or parasites. They are believed to be primitive relative of animals. There are four major types of protozoans. Amoeboid protozoans. These organisms live in freshwater, seawater, moist soil. They move and capture their prey by putting out pseudopodia. As in an amoeba, marine forms have silica shells on the surface. Some of them are entamoeba, are parasites. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic forms cause diseases such as sleeping sickness, for example, trypanosoma. Ciliated protozoans. They are aquatic, actively moving organisms. Because of the presence of thousands of cilia, they have a cavity that opens to the outside of the surface. The coordinated movement of rows of cilia causes water laden with food to be steered into the gut. Example is paramecium. Moving for sporozoans. This includes diverse organisms that have infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malarial parasite which causes malaria, a disease which has a staggering effect on human population. Now let's see the diagrams. Yes, wait, let me show you the diagrams. This is the diagram of paramecium. Yes, and this is a slime mold which is a saprophytic protist. These, what is this? This is euglenoid having one two flagellas, one long and one short, and they have a protein rich layer called pellicle. This is dinoflagellate, like this. Okay, so now moving ahead for kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and toadstool are also fungi. White spots seen on mustard leaves, they are due to parasitic fungi. Some unicellular fungi yeast. They are used to make bread and beer. Other fungi causes disease in plants and animals. Wheat rust causing Posinia is an important example. Some are the source of antibiotics. Example, penicillium. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent the food from going bad due to the bacterial and fungal infections. With the exception of yeast, which are unicellular, fungi are filamentous. Their bodies consist of long slender, thread-like structures called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. They are called sinusitic hyphae. Others have septae or cross walls in their hyphae. The cell walls of fungi are composed of chitin and polysaccharides. Important line. Most fungi are heterotrophic and absorb soluble organic matter from dead substrate and hence are called saprophytes. Those that depend on living plants and animals are called parasites. They can also live as symbiotes in association with the algae as lichens and with roots of higher plants such as pinus. as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spores. Now, spores, asexual spores are called conidia or sporangiospores or zoospores, and sexual reproduction is by oospores, ascospores, and basidiospores. The various spores are produced in distinct structures called fruiting bodies. The sexual cycle involves the following three steps. The first is fusion of protoplasm between two motile or non-motile gametes called plasmogamy, fusion of two nuclei called karyogamy, and then meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of two haploid cells immediately results in diploid cells. However, in other fungi, Ascomycetes and Basidiomycetes, an intervening dikaryotic stage, whereas N plus N, two nuclei per cell occurs. Such a condition is called dikaryon, and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and all cells become diploid. The fungi form fruiting bodies in which reduction division occurs, leading to the formation of haploid spores. Again, this is an important paragraph. The morphology of mycelium, mode of spore formation, and fruiting bodies form the basis for the division of kingdom into various classes. P by Q. Morphology of mycelium, mode of spore formation, and fruiting bodies form the basis of division of kingdom in various classes. Phycomycetes. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places, or as obligate parasites on plant. The mycelium is aseptate and cenocytic. Asexual reproduction takes place by zoospores or by aplanospores. Zoospores are motile and these are non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous or dissimilar. Some common examples are mucor, rhizopus, the bread mold, albugio, the parasitic fungi on mustard. Now coming to Ascomycetes, which is known as sac fungi. The Ascomycetes are mostly multicellular, penicillium example, or rarely unicellular example, yeast, Saccharomyces cerevisiae. They are saprophytic decomposers, parasitic or coprophilous, which is growing on dung. Mycelium. Mycelium is branched and septate. The asexual spores are conidia produced exogenously on special mycelium called conidiophores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asci. These asci arrange in different type of fruiting bodies called ascocarps. Some examples are Aspergillus, Claviceps, and Neurospora. This is can. We can remember this by can example. Yes. Okay, claviceps and neurospores used extensively in biochemical and genetic work. Many members like morels and truffles are edible and considered as delicious. Now moving to Basidiomycetes. Commonly known forms of Basidiomycetes are mushroom, bracket fungi, or puffball. They grow in soil, on logs, tree stumps, and live in living plants as parasites. Example: rusts and smuts. Mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent. Again, important line: sex organs are absent. Now, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis takes place in basidium, producing 4 basidiospores. The basidiospores are exogenously produced on basidium. The basidia are arranged in fruiting bodies called basidiocarps. Some common examples are agaricus mushroom, Ostolagos smut, and Posinias rust fungus on wheat. Yes. Now moving for deuteriomycetes. Commonly known imperfect fungi because only the asexual and vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and the sexual stage another. Later, when the linkages were established, the fungi were correctly identified and moved out of deuteriomycetes. Once the perfect stage, a sexual stage of members of deuteriomycetes were discovered, they were often moved to ascomycetes and basidiomycetes. The deuteriomycetes reproduced only by asexual spores known as conidia. Mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them they are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Colletotrichum, and Trichoderma. Now moving to kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic such as insectivorous plants or parasites. Bladder root and Venus phyta are examples of insectivorous plants. Cascade as parasite. The plant cells have a eukaryotic structure with prominent chloroplasts and cell wall mainly made up of cellulose. Planting includes algae, bryophytes, pteridophytes, gymnosperms, which we will study on later on. Life cycle has two distinct phases, diploid and haploid gametophyte, that alternate with each other. The length of the haploid and diploid phase, whether these phases are free living or dependent, vary among different groups in plants. This phenomenon is alternation of generation. Now moving to kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and they lack cell walls. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glycogen. The mode of nutrition is holozoic. That is by ingestion of food. They follow a definite growth pattern, grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. Now moving ahead to virus, viroids, prions and lichens. In five kingdom classification of Whittaker, there is no mention of lichens in some acellular organisms like viruses, viroids and prions. These are briefly introduced here. All of us have suffered the ill effects of common cold flu, known what effects virus can have on us. Even if we do not associate with our conditions, viruses did not find place in classification, since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having inert crystalline structure outside the living cell. This is diagram of what? This is the diagram of tobacco mosaic virus. Here is RNA present in a this is capsid. Capsid contain capsomere which protect RNA. This is what? Bacteriophage virus. Okay, now moving ahead for reading. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Virus means venom or poisonous fluid. Dmitry Ivanovsky recognized certain microbes as causal organism of mosaic disease of tobacco. These were found to be smaller than bacteria because they pass through bacteria-proof filters. These lines are very important lines. M.W. Bejernik in 1898 demonstrated that extract of infected plants of tobacco could cause infection in healthy plants and named the new pathogen virus and called the fluid as Contagium vivum fluidum. Infectious living fluid. ठीक है अरे ठीक है भाई. Okay, W.M. Stanley showed that viruses could be crystallized in crystal consist largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. No virus contain both RNA and DNA. A virus is a nucleoprotein and the genetic material is infection in general. Viruses that infect plant have single-stranded RNA and viruses that infect animal have single-stranded or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophage, they have what? They are usually double-stranded DNA viruses. The protein coat called capsid made up of small units called capsomere protects the nucleic acids. These capsomeres are arranged in a helical or polyhedral geometrical forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling, curling, yellowing, vein clearing, dwarfing, stunted growth. Now coming to viroids. Let me change the color first. Coming to viroids, we have viroids. In 1971, Theo Diener discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked protein coat and it is found in viruses, hence the name viroid. The RNA of viroid is of low molecular weight. All lines are important. All lines. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by agent consisting of abnormally folded proteins. The agent was similar in size to virus. These agents were called prions. The most notable disease caused by prion is spongiform encephalopathy, commonly called as mad cow disease in cattle, and its analogous variant Kat-Jacob disease in humans. Now moving for lichens. Lichens are symbiotic association, that is mutually useful association between algae and fungi. The algal component is known as phycopoint and fungal component is mycopoint, which are autotrophic and heterotropic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral, nutrients and water for its partner. So close is the association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicators. They do not grow in polluted areas. Again, phycopoint, mycopoint important for PYQs.